Hi guys, I'm Professor Brian Ives Araneta. Welcome to my microbiology thread. And here I will introduce you to the world of microbes. Now, microbiology is the scientific study of microorganisms. Here is a good visual showing the organisms studied by microbiologists. So those so-called too small to be seen by the naked eye. And we can group them into the cellular group here, the prokaryotic bacteria and archaeobacteria, and then the eukaryotic fungi and protists. And you have examples there. The acellular group include the viruses and similar particles like viroids, satellites, and prions. And these are technically dead, but they become active when inside of cells. And again, you might ask me, how about the macroalgae and mushrooms? They are obviously visible to the naked eye, right? So guys, that is the primary reason why we should not rely too much on the size of organisms studied in defining this field. So it's also useful to look at the techniques, usually lab techniques that are unique only to microbiology, because it can also be used to define this field. We have sterilization and aseptic transfer techniques. This is an autoclave. So it's used to kill microbes on any surface. So when you want to do sterilization. And of course, when you need to. Well, aseptic transfers, on the other hand, are needed to prevent contamination. For example, in this picture, the man is working on a virus, and I think he's transferring the sample to another container. And using this sort of chamber here, a biological chamber, for example, this can limit the chance that other microbes from external sources will contaminate the sample. Then we have molecular and pure culture techniques. Shown on the left is a DNA sequencer, and it is used to identify microbe samples after you compare the sequence with known sequences. For example, you may want to confirm if a suspected person has, let's say, COVID-19 virus. First, perhaps you need a sample or a swab or a tissue sample. Okay, this can be a useful guide here of these molecular techniques that will be used. Of course, it's not the actual procedure when testing for COVID-19, but again, it shows the basic principles when employing the molecular techniques. So first, you have to extract the DNA in the sample. So in the case of SARS-CoV-2 or COVID-19, it's an RNA virus, so we will be extracting the RNA. So again, the extraction of the genetic material is already a molecular technique by itself. Okay, of course, using special kits for RNA extraction. And after this, however, the amount that is extracted from the sample or from the original sample is still very little for analysis. So you need to amplify the genetic fragments using PCR. In this case, maybe they use some kind of reverse transcription or transcriptase PCR. Okay, again, this is another molecular technique. And then, once you have amplified the sample, that's a time when you can now sequence the sample. And the computer will just compare the sequence to online databases to compare it with the known COVID-19 virus. And so if it matches, then you're positive for the virus. Okay, pure culture techniques allow you to culture your microbe of interest in a culture medium. And in this picture, we have a plate loaded with a solid medium and colonies here growing on it. Now, pure means you grow only a single species and you avoid further contamination. And you can do all this with bacteria, fungi, viruses, algae. And also the media are varied and specific for the growth of a particular microbe you want to culture. But again, guys, you cannot culture every microbe. Many have, some have unique nutritional requirements that are hard to provide in the lab. Let's now go to naming and classifying microbes. Providing scientific names is is referred to as nomenclature. But again, classification is a different thing. When you classify, you group organisms based on the appearance or maybe based on their genetic relationship. So that's where the terms kingdom, phylum, okay, class, order, these terms are being used there in the classification. And here are some microbes and how their names were derived. So check it yourselves, guys, you can pause this video. Okay. Take note that we also adopt the two-name or binomial system used by Carl Linnaeus in the 17th century. Okay, this one is the three-domain classification system. It's the most recent and most accepted system. And as you can see, it tells us that all living things came from a single ancestor or origin. And that give rise to bacteria and then to archaeobacteria here. Okay, these simple prokaryotes are unicellular, mostly free-living with simple internal structure and metabolism. And then we have this third domain here, the eukaryotes, made up of advanced cells with a true nucleus. And usually the majority studied in microbiology include mostly the unicellular eukaryotes. Here are the major groups of microbes. I will also include the invertebrate parasites because they will also be studied in this field. Now let's have a quick look at the bacteria. So this is a picture of Staph aureus, the causative agent of acne, boils, including life-threatening diseases like pneumonia, meningitis, okay, and your toxic shock syndrome. And this is known to develop resistance to antibiotics. Again, bacteria has a simple structure. They are all unicellular, made up of separate individual cells. And they can also exist in various shapes, as shown here. Their genetic material is not enclosed in a nuclear membrane, so you cannot call it a true nucleus. And they only have a single set of chromosomes. That is, of course, prone to mutation. Because again, if you have a single set, you do not have a backup copy in case your DNA is damaged. Or unlike the higher forms like us, for example, we have an extra copy for DNA repair. That's why bacteria are prone to antibiotics. And their cell walls contain a unique material called peptidoglycan. In terms of nutritional types, they're also varied. They divide via binary fission. It's a form of asexual reproduction. Again, guys, I'm only providing a bird's eye view here in this presentation because, again, we will discuss these microbes in detail soon. Archaeobacteria are prokaryotic cells that live in extreme environments. So it's hard for us to imagine how these organisms live in these environments. For example, in hot springs, in areas where salinity is high, or in deep ocean vents where temperature and pressure is very high, or even in ice-cold regions. But it's not really required that they live in these harsh environments. You can also find archaeobacteria in the soil and other habitats, like, for example, in the digestive tract of ruminants, like your methanogens. But again, they were perhaps first discovered in extreme environments, so the description stuck. Next are the fungi. We have plenty of examples: molds, skin diseases, mushrooms. Under the microscope, they look like spherical bacteria, only larger, because they are now eukaryotic cells. You can also grow them in Petri plates like this one. But compared to bacteria, they grow slowly. You can get bacterial colonies in 24 hours, but with fungi, it can range from, let's say, 3 days to 3 weeks. And they get nutrition by digesting organic substances. So you name it. It can be rubber, wood, anything organic. And hopefully, they can also digest plastics. And again, they can also exist in two forms. The mold, as you can see here, it's an environmental form. And the yeast, when they are associated with hosts. Next, we have protozoa. Like the fungi, they are also eukaryotic but still single-celled. They are categorized based on their locomotory structures. In this case, cilia is the locomotory structure of the paramecium. Okay, so you note the hair-like projections on its surface here. There are also flagellated protozoans and the amoebas using pseudopods or false feet. And some are non-motile. Again guys, protozoans are one of the most diverse groups of organisms. Some are photosynthetic, while some can ingest bacteria or absorb nutrients directly. Therefore, they are heterotrophic. Okay, algae, let us limit to the unicellular ones. These organisms are easily observed in water. It makes the water look green or brown and yellow. And of course, it becomes more vivid in increasing counts. These are technically plants. So they can do some sort of primitive type of photosynthesis. They are the primary producers in the oceans. So they are eaten by small herbivores like krills and shrimps, and a variety of zooplankton, so they transfer the energy up to the top predators via the food web. And here we have viruses. Again, these are popular nowadays. We have SARS-CoV-2, popularly known as COVID-19 virus. Viruses are made only of DNA and protein that encases it. So they seem harmless as a particle, but they are infectious agents. Now, when contacting a receptor in their target host cells, they inject their DNA or RNA into the host cells, and they hijack the protein factory inside of the cells to produce more viral particles. And this will eventually lead to the bursting of the cell. And if the infection continues and damage is irreversible, so the host may die. And finally, we have these worms, okay, helminth parasites, invertebrates. They're also studied in microbiology because they cause diseases. And they are known to have unique life cycles that usually includes a variety of hosts, and their mode of infection are quite similar also to the microscopic microbes. Alright guys, so let us now look into how microbiology was established as a science. Let's have a bit of history first. Perhaps during the time when Ferdinand Magellan invaded the Philippines, the spontaneous generation theory is already a prevalent thought in Europe. Okay, this was made popular by thinkers and philosophers during that time. For example, flies can be generated out of rotten fruits. They believe that maggots can come from meat or fish. And there is even a recipe on how to make mice. So for example here, just mix grains with dirty clothes in a container, and after 21 days you will expect mice to just pop out. Now, this was already debunked or demystified as sort of fake news by a man called Francesco Redi, and this was his famous experimental setup. Here, maggots are present if the jar is not covered. Obviously, no maggots if covered, because of course, flies cannot enter. And in the third jar, when the cover was replaced by a gauze, the maggots were limited only in the surface, where flies lay their eggs. So this has already settled the issue. But when microbes were again discovered, the theory made a persistent comeback. This one is Antoine van Leeuwenhoek, or Leeuwenhoek. He's a Swedish merchant, a highly skilled lens maker, and maybe a self-proclaimed scientist. This is the microscope that he used, which is way advanced during that time. And the design, the concept is still being used in our compound microscopes today. Now on the right are his drawings of protozoa, bacteria, sperm, okay, things that he called as animalcules. And these are observable only under the microscope. Now in 1745, again, let's take a look at the comeback of spontaneous generation theory. We have John Needham. He claimed that even after he heated chicken broth and corn broth before pouring them into covered flasks, it becomes cloudy. And when placed under the microscope, it is teeming with microbes. So in short, Needham claimed that microbes develop spontaneously from the fluids. The point there is, if spontaneous generation is not applicable in large organisms, then right now it's occurring in microbes. And guys, it took 20 years later before this was challenged by Lazarus Spallanzani. Now, Spallanzani suggested that microbes in the air perhaps entered Needham's solution after they were boiled. So, Spallanzani showed that if you reheat the flask, it will not develop microbial growth. But again, Needham was firm and accused Spallanzani of destroying that so-called vital force that is necessary for spontaneous generation. Because again, he heated the broth and he also covered it, so the vital force was not able to enter. And we have several investigators who attempted to counter such arguments. We have Theodore Swan. He allowed air to enter a flask containing a sterile nutrient solution after the air had passed through a red hot tube, and the flask remained sterile. Okay, we have George Frederick Schroeder and von Dutch. They allowed air to enter the flask of heat sterilized medium after it had passed through a sterile cotton wool. Now, no growth occurred in the medium, even though the air had not been heated. You can check these experimental setups here, so you can pause again for a while if you like. But despite these experiments, there is this French naturalist, Felix Pochet or Pochet. He claimed in 1859 to have carried out experiments conclusively proving that microbial growth could occur without air contamination. And this claim provoked Louis Pasteur. So again, Louis Pasteur, he's a French chemist. He must settle the issue once and for all. So here, using a long neck flask, the broth was boiled and made sterile. Then while still hot, the neck was bent. That's why in some texts, they refer to this as one-necked flask. So, there's no excuse for poche here or poke. Air can therefore enter after the flask has cooled, and there was no microbial growth. The idea is obvious. So the microbes cannot move up along the bends here. So air can enter, but microbes are trapped along the bends. Then additional scientists also came up with simple experiments to support Pasteur's findings. We have physicist John Tyndall. He demonstrated that dust carried germs and broth remained sterile if air that entered does no dust. He also provided evidence for the existence of exceptionally heat-resistant forms of bacteria. Then we have botanist Ferdinand Kohn. He discovered that the heat-resistant bacteria recognized by Tyndall were species capable of producing bacterial endospores. So all of these findings, guys, lead to the development of aseptic techniques that we know are still being practiced until today. Now, in the next few slides, I will show you the various personalities that contributed to the establishment or sort of paved the way for this field of study to be recognized as a science. Now, let's start with the first golden age. I will present the most prominent ones here, and then you can pause this video to check the others from the timeline. Of course, depending on how we value each of them, all of these personalities are equally important in general. Now, first in line is Louis Pasteur. He has many other important contributions to microbiology aside from just debunking the spontaneous generation theory. As a chemist, he was once asked why beer and wine sometimes get sour. Now, this is the usual problem, especially if you transport large volumes of these beverages. And then you finally end up with sour wine with little to no commercial value. So Pasteur found out that yeast convert sugar to alcohol in the absence of air, and he called this fermentation. However, some microbes can also convert sugar into acids, like acetic acid or lactic acid, also using the same mechanism of anaerobic fermentation. So it turns sour. And so Pasteur suggested to heat the juice first to kill the unwanted fermenters before adding the yeast. And this process was called pasteurization. So it's a process also named after him, of course. And make sure that the heating should be enough to kill the microbes, but of course not to destroy the drink. Or, again, this is also common in milk. So maybe 60 to 70 degrees Celsius, okay, but not more than 100. And he also won international acclaim in 1885 with the discovery of the anti-rabies vaccine. We have a 9-year-old boy who had been bitten by a rabid dog. He was brought to Pasteur and was injected many times with increasingly virulent preparations of the attenuated or weakened virus. And the boy survived. Okay, and he also developed a vaccine against cholera in hens, against anthrax, and against diamond skin diseases in pigs. Okay, and so for this, people from around the world contributed to the construction of the Pasteur Institute in Paris, France. And one of the initial tasks of the institute was vaccine production. So basically, Pasteur was among those who established the germ theory of the disease. Again, what is this germ theory of the disease? So it's the idea that diseases are caused by specific microbes. Again, during that time, it was thought that it's of supernatural origin, maybe caused by witches or a curse. But again, it was somewhat established that diseases are caused by microbes. And also, when Pasteur died, he left plenty of research works, which were of course continued by his followers. Then an English surgeon named Joseph Lister came up with a strategy that minimizes wound infection. So inspired by Pasteur, he developed what we now call as antiseptic surgery. Okay, so all surgical tools were disinfected using phenol, and physicians and patients observed proper hygiene. And since it increased the success rate of surgeries, it was also adopted by other surgeons. And up to this day. Then we have Koch, a German physician, another big man in microbiology. And the first direct demonstration that bacteria causes diseases was his work on anthrax. And Robert Koch actually made that during the time when he and Pasteur are racing for a cure against anthrax. Okay, so here he established what we now call as Koch's postulates. So it's a sequence of experimental steps for directly proving or relating a specific microbe to a specific disease. So first, the disease agent must be present in the tissues of the affected organism. So here, Koch developed a way to detect the tuberculosis bacteria in the tissue. Okay, second step is you should be able to isolate and grow it in a culture medium. Then the resulting colony, when injected into the healthy organism, will still cause the same disease or symptoms. And you will also be able to isolate or detect the same microbe from the second person or organism. So definitely in this timeline, Koch was credited also to discovering TB or tuberculosis. It was also in his lab where pure culture techniques were developed. So prior to the use of agar, they used potato as a solid medium. But then it's not enough to support a wide range of bacteria, especially those with unique requirements. So it is needed to make artificial media, and you should be able to solidify it. So first they use gelatin, but it melts when sterilized at higher temperatures. And it was Fanny Hess who suggested the use of agar. And of course, agar is more stable at higher temperatures. Hess was actually the wife of one of Koch's lab assistants, Walter Hess. And then the Petri dish was also fabricated to ensure aseptic transfers and to isolate cultures from further contamination. Also, the discovery of Vibrio cholera was attributed to Koch here. Now, this guy is also worth mentioning, even though he was not on the timeline here, because he existed before that. So Edward Jenner is an English physician who developed the first vaccine for smallpox. So meaning prior to Koch and Pasteur, vaccination is not new. Jenner observed that milkmaids exposed to cowpox are relatively resistant to smallpox. So he thought, why not inject cowpox to healthy individuals? And it will cause perhaps a mild fever. Then the immune system will produce antibodies and memory cells that will combat smallpox. So in short, it worked. So the term vaca from cow was used to refer to vaccination. So in this painting, actually, it was said that Jenner was convincing the farmer to have his son vaccinated. Okay guys, so just pause the video if you need to look at all the personalities in this timeline. After all, we will still encounter them in future discussions. Again, a lot of disease-causing microbes were discovered. Then some personalities here were also able to discover the role of microbes in nutrient cycling and immunity. And even a tumor-causing virus was also discovered by Rouse. And this won a Nobel Prize. Now in the second golden age, as you can see here, we can see the rise of chemotherapeutic drugs. Now, treatment of disease using chemical substances is called chemotherapy. And the first antibiotic was discovered by accident by Alexander Fleming. If you know his story, he was about to throw away expired plate cultures when he observed that a particular mold was able to inhibit the growth of bacteria. So meaning there must be a substance produced by this mold that is lethal to the bacteria. And this was later purified and put into clinical trials. And it was successful, of course. Now, since these early discoveries, thousands of other antibiotics have been discovered. Waxman discovered streptomycin. Then the steps of the Krebs cycle was also elucidated. Then poliovirus was successfully cultured in cell cultures. Again, just pause and take a look at all these personalities. Special mention goes to Walter Gilbert, together with his graduate student, Maxam, and Frederick Sanger. In 1977, they worked separately. Gilbert was in the United States and England. Okay, they developed new techniques for rapid DNA sequencing, and the methods devised by Sanger and Gilbert made it possible to read the nucleotide sequences for entire genes, which of course can be as long as 30,000 bases. Now, for discovering these techniques, Gilbert and Sanger received the Albert Lasker Medical Research Award and shared the Nobel Prize in Chemistry in 1980. Now, the third golden age is where we now have advances in molecular genetics. Okay, then we have new DNA sequencing tools, and computers now allow investigators to study all the DNA in an organism. So that can help them identify genes and their functions. And there was this field called genomics, so the study of all the organism's genes. Now, today, scientists are able now to classify organisms according to their genetic relationships with each other. And most of the studied organisms are microbes. Also, we have microbes that are genetically modified to produce useful gene products. There is what we call now as recombinant DNA technology, which inserts, for example, the DNA of another organism into bacteria. And it's now the bacteria that can produce the desired protein product. Now, the development of this technique, your recombinant DNA technology, genetic engineering, this has revolutionized research. And of course, practical applications in all areas of microbiology. Alright guys, so let's now have a look at how microbes have influenced our society, or shall we say how they become useful to us humans. Okay, there is a field called microbial ecology due to the works of two microbiologists. You will encounter them soon, Bejirink and Winogradsky. Now, the role of microbes in nutrient cycling is already established. As you can see in this picture, most microbes are the decomposers. So they return all the elements taken up by once live organisms back into the environments. Without this, of course, we will have piles of dead bodies, plant material. You can also see here how nitrogen gas enter the biological systems, and we really owe our nitrogenous substances from microbes. Okay, here, of course, sewage treatment may not be common here in the Philippines, but of course in first world countries, in first world cities and urban centers abroad, they retrieve wastewater back by treating sewage. Okay, sewage may be composed of wastewater, runoff, okay, industrial waste, and it is basically 90% water or 99% water. The rest are suspended solids. So maybe we can use microbes to try to metabolize and remove the organic matter content or toxins. Or we can also remove harmful microbes from sewage. We will look at the process later. Okay, bioremediation is the use of any organism to clear up pollutants. And that's what they do with oil spills, for example. So here is a picture before and after the spill. So after introducing microbes and enriching the environment with nutrients, the oil metabolizing microbe cleared up the oil and converted it back to carbon dioxide and water. Okay, this is one method on how to utilize candidate microbes to clear up pollutants. So first you have to do in vitro simulation, and then if the microbe of interest can remove the pollutant, you can introduce it into the environment. Okay, pest control. The bacterium Bacillus thuringiensis has been used extensively in the US to control pests. So it's incorporated in it's usually in a powder form. It's applied to crops, so that when insects eat this, they die, because the bacteria again produce protein crystals that are toxic to the digestive system of these insects. And using recombinant DNA techniques, this gene has already been inserted into some plants to make them insect resistant. So if you recall BT corn or BT eggplant, these are genetically modified organisms that can produce the BT toxin that is lethal to pests. And we can look at biotechnology here as the practical application of living organisms. Of course, we will be using here microbes. Okay, we will use microbes to produce something of commercial value. So best example here is of course fermenting alcoholic beverages. But again guys, there are plenty of other applications. So you name it, genetic engineering, gene therapy, processing of food products like cheese and yogurt for example, or production of insulin. So those are just one of the applications of biotechnology using microbes. And finally, of course, the role of microbes in human diseases has been established since 17th century. And we now know a variety of causative agents to lots of diseases. And right now we are also looking at the role of our normal microbial flora. This field is now gaining popularity because again, the future of our health, as they say, relies on the bacteria that lives on us. So it's actually estimated that microbes outnumber the cells in our body, like 10:1 or maybe 2:1. Here we'll also be discussing antimicrobial drugs, vaccines, okay, diagnostic tests, and how diseases spread. And that's for now, guys. I'll see you again next time. Again, thank you very much for watching. Don't forget to like this video and subscribe to my channel.